welcome Dr. Martin Oberbarnscheidt from the University of Pittsburgh. So Dr. Oberbarnscheidt received his MD and PhD degrees from the Free University of Berlin. He then moved to Yale to do postdoctoral research on cell organ allograft rejection with Fadi Lacus, and then moved to the University of Pittsburgh, where his work focuses now on the role of innate allorecognition in transplant. So this is an area of transplant immunology that's been previously a bit overlooked, but recently he's actually made really important contributions to understanding how innate immune cells modulate the pro or anti-inflammatory environment. So Dr. Oberbarnscheidt has made exciting contributions to this rapidly developing area, which I'm sure he'll talk about this today, including a recent discovery of a mechanism of allorecognition by the innate immune system. So with that, I'm going to turn the presentation over and look forward to the talk. Thank you so much.
Thank you very much for the introduction, and it's a pleasure for me to be able to present our work to you today. And I am hoping that we can make this interactive. And I would encourage you to interrupt me and ask questions whenever you have any, and not wait till the end. So let me try to get things going here by. I need some permission, I think, to share my screen. And then we can get started.
Maybe Laura is the host. I think that maybe Laura can...
Yes, no, it's fine.
Laura might have to do that.
Let's see.
Perfect.
All right, so now the big question always is, what are you seeing? Are you seeing the slide, or are you seeing the presenter view?
We see the slide now.
Okay, great. And let me try to move this out of the way. Okay, great. So today, I'm going to tell you the story of our work over the past, well, more than 10 years almost, and that is about innate allorecognition. Okay, here is my mouse. Can you see my mouse pointer? Let me try change that to a laser pointer. Okay, so when we talk about non-self that our immune system recognizes, we can divide it up into two major groups. One is microbial non-self, and this includes all the pathogens that we're exposed to. And the other half that gets neglected frequently is allogeneic non-self. And these are transplants, allografts, but there are also fetuses and tumors that develop in the body. So if we think about sensing non-self, at the center of our immune system is the adaptive immune system with T and B cells, with a variety of T and B cell receptors that can undergo somatic hypermutation and be very specific in reacting to non-self. And connecting this with our environment is the innate immune system. This is where we sense non-self. And this is primarily happening through pattern recognition receptors. And we know fairly well this is how we respond to bacteria, viruses, fungi, and parasites. And one of the representative cells that is able to bridge the innate with the adaptive immune system is the dendritic cell, professional antigen presenting cell that can sense non-self, microbial non-self, for example, get activated, mature, and then activate the adaptive immune system. But when we think about transplantation, I see. I have to, maybe I can use the arrows. There you go. When we think about transplantation, the big question we had early on was, what is the adjuvant for allografts? When we have transplant antigen, what is activating the innate immune system and then the adaptive immune system? We have known for a long time that T and B cells are a big factor in causing allograft rejection, but what exactly activates the innate immune system was not entirely clear. So one of the hypotheses was that it is just microbial contamination along with the transplant procedure, or just the release of danger signals that cause the innate immune system to get activated. So we designed a very simple experiment that would allow us to test this hypothesis that if it is just microbial contamination or danger signals, then comparing the innate immune responses against allo or syn grafts should be identical. So the only difference is danger, which should happen via a surgical procedure, or microbial contamination. It should happen with both allogeneic grafts and syngeneic grafts. And when we performed this experiment, we found out that the allo response does not equal the syngeneic response. So we did this with heart transplant. We transplanted heart allografts into RAC gamma chain knockout recipients, syngeneic ones and allogeneic ones. And the difference we found in infiltrating immune cells in the allograft was that of mature monocyte-derived dendritic cells. So these are CD11b positive cells that are F480 negative, and they express class 2 and CD11c. And we found an increase here early on after transplantation, but a prolonged maintenance of monodisease in allografts, but not in syngeneic grafts. So in syngeneic grafts, there was a fairly rapid decline, and then they disappeared. We followed those mice up until day 42. And when we did this in kidney transplants, we found a fairly similar thing, that in allografts, here the recipient monocytes are colored in green. This is intravital imaging of a kidney graft. And you can see that in the allogeneic graft, there are many more green cells, and this is GFP expressed under the CX301 promoter, than in a syngeneic graft. And this is accordingly the quantitation. Now, these monocyte-derived dendritic cells in allogeneic grafts would produce IL-12, both at early and late time points. And when you put them together with T cells, these T cells would produce interferon gamma, both at early time points and here also shown at late time points on day 42. And this is just the control with LPS stimulated splenocytes. So in summary, we have shown that monocyte-derived dendritic cells infiltrate allogeneic grafts more than syngeneic grafts after transplantation. And only in allogeneic grafts, they go on and produce IL-12 and generate a Th1 T cell response with interferon gamma production, leading to graft rejection. Now, when we took a closer look at these dendritic cells and monitored donor-derived dendritic cells versus recipient-derived mono-DCs, we made the observation that the donor DCs quickly disappear after transplantation. And this is a kidney graft that I'm showing you here. This is before transplantation. You can see all these yellow dendritic cells of the donor. And this is showing you day 3 micrograph of a kidney graft, where you can see occasional green cells appearing, and then the yellow cells disappearing. And by 7, you can see heavy infiltration with recipient-derived mono-DCs. Now, we were asking, what are these mono-DCs doing when they infiltrate these allografts? And one of the functions that we were able to elucidate by intravital imaging, looking at colored T cells and colored monocyte DCs in the graft, is that mono DCs are responsible for I think I have to switch back to replay this. They're responsible for causing T cell migration through the capillary into the graft tissue. You can see here the red T cell that is contacting the mono DC and then extravasating into the graft tissue, where it's then still in contact with the dendritic cells and it can exert effector function. Now, what role do these mono-DCs have in rejection? One of the first experiments we did was try to deplete monocytes or dendritic cells and monitor graft outcome in terms of histology. So what I'm showing you here is CD11B cell depletion after transplantation. So we were transplanting a heart graft in this case, an allogeneic heart graft into either wild-type chimeras or CD11B DTR chimeras and treated with diphtheria toxin for 7 days, then harvested the grafts and looked at T-cell infiltration. And as you can see, the wild type has lots of red colored T cells infiltrating the graft tissue, whereas the allogeneic graft in the CD11B depleted mice shows a lot less infiltration. Now, one of the reasons, as you will probably know, why we only did this short term and that you get to see histology is that the CD11B DTR mouse requires very high doses of diphtheria toxin, so that despite creation of bone marrow chimeric mice, these mice, even the wild type chimeras, would die after about 10 days of high dose diphtheria toxin injection. So we went on to deplete CD11C cells after priming had already happened. And this is also heart transplantation. We're using bulb-C into B6. And again, we used chimeric mice here, either CD11C DTR or wild-type bone marrow chimeras that were treated with diphtheria toxin starting on day 5 after transplantation, allowing initial priming in the lymph nodes to happen so that we would have activated effector T cells before we start depleting all dendritic cells, as this is a systemic treatment. Nevertheless, you can see that the wild-type grafts are rejected quickly, whereas we see pronounced prolongation of heart allograft survival in the CD11C depleted recipients, indicating that mono-DCs have a role in organ rejection. Now, one of the big questions we asked next was, what are these monocytes sensing? I've shown you the process where monocytes mature into dendritic cells who are then presenting antigen and can activate T cells and lead to rejection. But what really is happening here at the beginning? And to study this, we screened a large number of donor strains and looked at infiltration with mono-DCs as the readout. And again, as the recipient, we used B6 RAC gamma chain knockouts devoid of any lymphoid cells. And when you compare the response to these different strains, some are wild-derived, such as the Pahari mice or the cast mice. Some are known inbred strains like the C3H or the NOR and NOD mice and the bulb-C mice. You can see that the innate response to the various donors varies significantly. And one of the observations we made was that there is a large discrepancy between the response to a NOR graft and a NOD graft. So what I would like to highlight, because I haven't mentioned it before, is that these transplants are bone marrow plug transplants. They are a very fast way to transplant allogeneic tissue, and you push out the intact bone marrow plug out of the femur of mice, and we transplant it under the kidney capsule. And then we can look at cell infiltration at various time points after transplantation, usually 1 week after the transplant procedure. Now, as you all know, the NOD mice are the non-obese diabetic mice, and the NOR are the non-obese diabetes-resistant mice, and they vary in their innate response of the host to them quite significantly. So to further study this, we transplanted 2 bone marrow plaques into the same recipient under opposite kidney capsules. And we were then able to compare 2 plugs, the infiltration of 2 plugs in the same recipient. So here you can see that there is not much of a difference between the B6 syngeneic and the NOR bone marrow plaque transplant. But you can see that if you transplant the NOR and the NOD in the same recipient, you see a large response mirroring what we found when we did individual transplants. Now, the difference between NOD and NOR mice is fairly small. The NOR mice contains 88% of its genome from the NOD mouse, and the rest, the majority of them, is derived from B6 mice. And in collaboration with Jane Danska, who had created congenic NOD and NOR mice, we used these congenic mice to figure out where the difference between the response, the different responses is. And the two congenic mice I would like to highlight is the R7 mouse and the R12 mouse. The R7 mouse is a NOD mouse that contains a segment of NOR, and this is all within the IDD13 segment. And within that segment is the SERP alpha gene. Likewise, the R12 contains a piece of the NOR genome, which is B6 in this case, that is not in the SERP alpha area. And when we compared the responses of R7 and R12 in the host and their infiltration with mono DCs, we saw a severe reduction in the response in the R7 mice, which house the SERP alpha of the B6, which was not the case in the R12 congenic, which has the original NOD SERP alpha receptor, indicating that one of the possible genes of interest could be SERP alpha in this response. Now, again, when we compared the bone marrow transplants of NOD and R12 within the same recipient, we could detect no significant difference between the responses to the allograft. And here, finally, on the right side, I am showing you the response to now NOD RAC gamma chain donors or B6 RAC gamma chain donors. And the donor here in the middle is a B6 RAC gamma chain knockout that contains a very small gene segment of about 10 genes, one of which is the NOD SERP alpha. So here you have a mostly syngeneic mouse that has the NOD SERP alpha. And as you can see, you now can elicit a response that is almost indistinguishable to the NOD RAC gamma chain response and significantly higher than the syngeneic response to B6 RAC gamma chain knockouts. Okay, so SERP alpha, donor SERP alpha is one of the allodeterminants. So how do we think this works? We think, as you know, SERP alpha and CD47 are commonly thought as the do not eat me signals. CD37 is present on all nucleated cells and on RBCs too, on all cells. And monocytes have a SERP alpha on their cell surface as well as CD47. So here we're saying that the balance of sensing SERP alpha on a self cell as well as CD47 is resulting in a positive and negative signal that results in a net zero response, which leads to no response because it is self. If we have non-self, we're hypothesizing that now you get a larger positive signal through CD47 due to the allogeneic SERP alpha on the donor. And you still get a negative response because SERP alpha on the monocyte is detecting CD47 on the non-self cell. And if you block this interaction specifically between the SERP alpha on the donor cell and CD47 on the monocyte, the recipient monocyte, you get no response. And the way we have done this is we have treated mice with human CD47 FC or with a human IgG1 control FC What the human CD47 FC does is that the NOR-SERP alpha has a very high affinity for the human CD47, contrary to the B6 SERP alpha. So using this human CD47 fusion protein, we could specifically block NOD-SERP alpha on donor cells. And as you can see in the quantitation of infiltrating mono-DCs here, you get a suppressed response because you block this interaction. Likewise, if you take away CD47 of the recipient, you also get less infiltration, indicating that interrupting that SERP alpha CD47 pathway between monocytes and other cells is important in sensing allogeneic non-self. Are there any questions so far?
No.
Okay.
Can I ask a question? So is the SERP alpha polymorphism also present in humans?
That's an excellent question, and I have one slide to show you, and in 2 slides that will address that.
Okay.
But the short answer is yes, it is polymorphic.
Perfect. Thank you.
So a question about the balance of the SERP alpha signal and the CD47 signal. So what are the signaling pathways for those molecules?
Yes, that's also an excellent question, one that we have not really investigated further. So this CD47 signaling, so for the longest time, CD47 was viewed as a receptor that would be providing a signal to SERP alpha, and not a receptor that would do active signaling itself. There are some hints in the literature that CD47 can signal, but the pathways are not well understood. And we have not done any further follow-up experiments to elucidate the signaling that happens upon encountering allogeneic non-self. So I can't really tell you how the signaling works. But what we did is we looked at the effects of the CD47 SERP alpha on rejection. And this is work done by our talented microsurgeon, Da-Chang Zhao. He's unfortunately back in China, but he transplanted kidney grafts, and kidney transplantation in mouse models is in the majority of strain combinations, a chronic rejection model. So here he transplanted either syngeneic grafts, so meaning B6 to B6, or NOD to wild type, or NOD to CD47 recipients. And what you can see looking at the histology here is if you compare the syngeneic with the allogeneic graft, you see a lot more nodular infiltration, and you see fibrosis here in blue, indicating chronic rejection. And if you compare that with the histology where we transplanted the same allogeneic graft into CD47 knockout recipients, you can see that the grafts look almost pristine. And this is reflected in quantitating the nodule-like infiltration areas, where you can see a significant reduction in the nod to CD47 knockout recipient transplant, and also in the amount of fibrosis that is present, where here the nod into CD47 knockout recipient is
it me? I think that there is a problem with microphone.
Yeah, I couldn't hear you very well.
Just a few seconds ago, I started.
See.
Yeah.
Okay, can you hear me?
Yeah, much better.
Okay, I just re-plugged it in. Sorry for that. Okay, so I don't know where I lost you, but I was mentioning that the infiltration area in the CD47 knockout recipients were similar to the syngeneic transplants, as well as the fibrosis, the area of fibrosed tissue in the CD47 recipient, knockout recipients compared to the syngeneic grafts. So you're basically able to convert in this specific model an allogeneic graft into what looks like very similar to a syngeneic graft. Likewise, when you get rid of CD4 of SERP alpha in this case, and here we're utilizing the a NOD B6 SERP alpha mouse. So this is a NOD mouse that expresses just the B6 SERP alpha. So everything is allogeneic compared to the B6 wild type recipient, except that the SERP alpha is syngeneic in this case. And again, you can see that infiltration in the strain combination is very similar to what you see in the CD47 knockout recipient and much lower than in the wild type transplant. And the same is true for the fibrosis area. Now, does it have any relevance to humans? We don't really know yet, but what we looked at is at the 1000 Genome Project, and there are more than 100 haplotypes of SERP alpha. And we were able to, or we listed here the 13 most common ones according to their ethnicity. So you can see that most humans have haplotype number 1 of SERP alpha, and then there is some variations in which haplotypes are the most common and what percentage of the population will have those in other populations. So these haplotypes we can identify by PCR, and we can do functional analysis and potentially disease correlation. And we're studying this in healthy populations, as well as kidney transplant recipients and bone marrow transplant recipients. But this is all I can tell you about this, as I don't have any further results at this point to show to you. So one of the other questions we had is if monocytes can acquire allospecific memory. And the reason why we were interested in this was during our initial experiments that looked at ear swelling upon injection of allogeneic versus syngeneic splenocytes and in RAC knockout mice. We found that there seemed to be a heightened response after initial priming. So we studied this again in a bone marrow transplantation experiment. And we immunized RAC gamma chain knockout recipients with either no immunization, B6, BALB/C, or C3H splenocytes that were irradiated, and then transplanted a BALB/C bone marrow plug after 7, 28, or 49 days, and came back 7 days later to analyze graft infiltration of the bone marrow plug. And what you can see in this graph is that only upon recall with a BALB/C graft after a prior immunization with BALB/C splenocytes do you see a heightened response. So if you immunize with syngeneic B6 or with third-party C3H splenocytes, you do not get a heightened response, and that is true for day 7 and day 8 after primary immunization. Around day 49, we found that we could not detect significant differences anymore, potentially indicating that the memory response we saw here is fairly short-lived, at least when you compare it to adaptive immune cells. Now, we turn the experiment around and now challenge with the C3H grafts. And again, you can see that the response after C3H immunization and only after C3H immunization is heightened when you come back with a C3H graft, but not with the other immunized splenocytes. Now, to make sure, because here we were immunizing with wild-type splenocytes that were irradiated, to make sure that that this is still happening in the setting where no lymphoid cells are around, we this time used splenocytes of mice that were all on a RAC gamma chain knockout background. So here we had to switch strains slightly because not all strains are available on a RAC gamma chain knockout background, but we used B6 syngeneic, BALB/c, and NOD splenocytes from mice on a RAC gamma chain knockout background. And again, you can see that only when we immunize with BALB/c, RAC knockout, gamma chain knockout splenocytes, we could elicit a recall response upon transplanting a BALB/c graft. Now, to convince you that the NOD, or the response to NOD RAC gamma chain knockout bone marrow plugs is still a sufficient response, and there is not something wrong with using these at donor mice, we included a control where we immunized and recalled with not RAC gamma chain knockout splenocytes. And you can see that we were able to elicit a heightened response in that model, in that strain combination also. So to further prove that it is really the monocytes that become memory, we did an adoptive transfer experiment. So similar to the previous experiment, we immunized mice with either syngeneic or allogeneic splenocytes, and then came back 7, 21, or 35 days later, sorted the monocytes from the spleen, and then adoptively transferred them into a new B6 RAC gamma chain knockout recipient, transplanted the BALB/c bone marrow plug graft, and then 7 days later came and analyzed the graft. And again, you can see that we see heightened responses upon transferring allogeneic monocytes, or monocytes from allogeneic immunized mice versus the syngeneic ones. And this is true this time across all the time points up until day 35. So now we were interested in figuring out what is responsible for their memory response. What are monocytes sensing here? And one of the observations we made is that an MHC mismatch is necessary for memory induction and recall. So again, we did a series of priming and recall experiments, starting with the primary response shown here in black, then the prime boost of BALB/c showing the response in red here. And then if you come and prime with a bulb.b mouse that has the B6 MHC, but the rest is all BALB/c allogeneic, and then come and recall with a BALB/c, you see that the memory response is diminished and fairly identical to the primary response to BALB/c grafts. Okay, so likewise, if you prime with BALB/c, but then you come back with a BALB/b bone marrow plug where there is no MHC mismatch, you also see a response that is in line with the primary response to BALB/c bone marrow plugs, indicating that MHC mismatches are necessary for priming and recall. So we did similar experiments in a system where we used macrophages. So our collaborator Sean Lee had shown that if you immunize B6 RAC gamma chain knockout mice with allogeneic splenocytes and give anti-CD40 after 7 days, you can observe killing mediated by these macrophages of allogeneic splenocytes that are transferred IV. And this is a standard CFSE killing assay where you transfer two different cell populations that are labeled with different concentrations of CFSE, and you can see here the naive control. So without immunization, there is no killing of BALB/C allogeneic splenocytes. If you immunize these mice with BALB/C, you can see that BALB/C splenocytes are efficiently eliminated. And when you transfer class 1 knockout splenocytes, you can see that killing is impaired, and this is reflected here or quantitated here in the graph. And likewise, using BALB/B targets where the MHC is matched also resulted in less killing of these otherwise allogeneic splenocytes, indicating that class 1 recognition is necessary for memory generation. And when we screened databases for receptors that are expressed on monocytes and macrophages, we stumbled upon paired immunoglobulin-like receptors. In the mouse, these are peers, and the human equivalent are the LILRs. And both receptor groups have activating and inhibitory receptors. And in the mouse, these are expressed on macrophages, mast cell granulocytes, DCs. PIR-B is expressed in B cells. And the human equivalent is fairly similar. Okay. So we tested if monocyte sensing of non-self MHC is mediated via peer receptors. And in order to do that, because the reagents are fairly limited, we used a PIR-B knockout recipient that was bred on the RAC gamma chain knockout background, and then treated either with antibody or with anti-PIR-AB antibody. So the anti-PIR antibodies, there's really only one that recognizes both PIR-A and PIR-B. In the mouse, there's only one PIR-B receptor, but 5 PIR-A receptors. And there are no specific PIR-A antibodies that would recognize just subtypes of PIR-A or PIR-A in general. So doing this, we eliminated PIR-B, and so that when we would give an anti-PIR-AB antibody, it should block only the PIR-A. And as you can see here, the memory response was completely abrogated after treatment with anti-PIR-AB antibody. Also, when we treated mice with a PIR-A3 FC fusion protein after immunization and recall with BALB/C, we saw that we could block the memory monocyte response completely. If you do the same in the C3H system, priming and recalling with C3H, you can see that here PIR-A3 was not able to block the memory response to C3H, indicating that PIR-A3 is fairly specific to blocking the response to BALB/C class 1, but not to the C3H class 1. To further test this, we also generated PIR-A knockout mice, and these were also on a RAC gamma chain background. And you can see that the memory response of PIR-A knockout mice is diminished in response to BALB/C, both on day 7 as well as later on day 28. So it's not a defect of monocyte priming, because the primary response here of the PIR-A knockout and the wild type mice are similar. It is truly a defect in the memory response, both at an early and at a late time point. So, of course, we were interested in figuring out if there are any effects of PIR-A on chronic rejection. So we utilized these mice, the PIR-A knockout and the PIR-B knockout and fusion protein treatment, to study what happens to renal allograft survival after transplanting BALB/C to B6 wild-type recipients. So we made two observations here. As I told you, the kidney transplant model is a chronic rejection model. So the wild type donors and recipients lead to prolonged allograft survival. If we transplant a biopsy into PRA knockout recipients, we also got prolonged allograft survival. So that's not different from the wild types. But when we transplanted into PRB knockout recipients, so PRB is the inhibitory receptor, we observed fairly early allograft rejection. And this was also the case when we transplanted into PRB knockout recipients and treated with a control IgG. But when we added the PRA3 FC fusion protein, so remember now this is blocking the PRA3 class 1 interactions, we also observed prolonged allograft survival, indicating that there is a role for PRA in chronic renal allograft rejection. And this is also reflected in the histology. You see again nodular infiltrate and fibrosis here, and upon transplantation in a PRA knockout recipient, you see that the grafts look fairly pristine. The same is true for a heart transplant model. Now, the heart transplant model is an acute rejection model. So here in the mice that you don't treat with anything, you see fairly quick rejection around 9 or 10 days after transplantation. And again, when you treat these mice with PRA3FC, you see prolongation of allograft survival, as you do in a commonly used model of co-stimulatory blockade with CTLA-4 Ig. And when you combine those two treatments, we were able to get long-term cardiac allograft survival. And again, the histology shows a reduction in fibrosis and infiltration in these allografts, indicating a good survival and function. Now, to further investigate the mechanisms of monocyte memory, we performed single-cell RNA-seq. And what I'm showing you here is a pseudotime analysis where we were able to identify distinct states of monocytes. The S state we designated as starting state, and then we have these expanded states, E1, E2, and E3. And when we specifically looked at the expression of PRB and PRA2 and PRA3, these were the peers that we found transcripts for in this experiment, and compared the expression between those different states. In all of these expanded states, we noticed an upregulation of PRA2 or PRA3 compared to the starting state, indicating that these monocytes potentially expand. And when you look at proportions of cells in these individual states after immunization with different splenocytes, we observed that in the BALB-C and the C3H immunized monocytes, you saw an increase in monocytes that were in the E1 through E3 states compared to the syngenetically immunized mice. And then finally, when we looked at the expression of either PRA2 or PRA3 or expression of both in those four states, we observed that there is very little, there are very little cells that express both of the PRA molecules, indicating that there could be variegated expression of the PRA receptors on monocytes. And to further investigate if these monocytes can clonally expand after immunization, we used tetramers specific for H2KB, D, or K, immunized with the different the donor strains that we have used previously, the B6, syngeneic, BALB-C, and C3H. And we used wild-type recipients, pure B knockout recipients, and pure A recipients. So if we first look at the wild type, you can see that what I would like to highlight is that upon immunization with BALB/C and staining with a D tetramer, we saw an increase in the monocyte population that expresses the PRA3, because we had shown that this is specific for the D tetramer. The cleanest result we obtained in the PRB knockout mouse. So here, and this is gated on monocytes. So here we're eliminating binding to PRB, and potentially or ideally are only staining PRAs, and you can see a nice increase in monocytes staining for the D tetramer after BALB/C immunization. And likewise, if you immunize with C3H, you see the tetramer, the K tetramer increasingly binding to a larger proportion of monocytes. And to confirm that these tetramers are really binding to the PRA molecules, we use the PRA knockout as recipient. And you can see that we find very little to no binding irrespective of which cells we immunize with, indicating that we see clonal expansion of monocytes after encountering a primary immunization. So we think what is happening is that specific monocytes recognize allogeneic non-self in form of their class 1 molecules, and this then leads to a clonal expansion of memory monocytes that then can mediate a heightened response to allogeneic non-self. So now we're going back to SERP-alpha. What does SERP-alpha have to do with innate memory? Is there any link, and does it matter? So what I'm showing you here is the response to NOD again. And here in the blue dots, we changed the immunization to the NOD B6 SERP-alpha mouse. So everything is allogeneic, including the MHC, but the SERP-alpha is syngeneic. And then when we recall with a fully allogeneic SERP-alpha mismatched NOD, we cannot elicit a memory response, indicating that SERP-alpha differences are necessary at the priming stage to form memory. On the other hand, if you look at the response to NOD with a B6 SERP-alpha, you see a baseline response here that is fairly low. But if you immunize now with a fully allogeneic SERP-alpha mismatch NOD splenocytes, and then come back and recall with the NOD bone marrow plug that has a syngeneic B6 SERP-alpha, you can still elicit a memory response. So in summary, SERP-alpha mismatches are necessary to generate innate memory responses, but they're not necessary for the recall response. And then again, if you get rid of CD47 in the recipient, you cannot elicit any response at all. Okay, so what do we think is happening? We think that both are needed, that monocytes sense donor SERP-alpha that is polymorphic and via their CD47 receptor, and that they also sense allogeneic class 1 through their PRA receptors. And this leads to the primary response by monocytes maturing to dendritic cells and initiating a T cell response that can lead to rejection. And it also leads to the generation of memory monocytes. So when we look at the model that I showed you initially, we can now add innate allorecognition to this. And I hope to have shown you that there is specificity in the innate allorecognition. There is memory, although if you compare it to the adaptive memory, it's more short-lived, but it is in line with what people have seen for NK cell memory. There is clonal expansion, and there is diversity because these receptors can detect diverse non-self. So what are the clinical implications? The clinical implications, we think, are that you have to be aware that the innate immune system will continue to sense non-self in the allograft, potentially contributing to chronic rejection and or preventing tolerance. And it's also a very attractive potential therapeutic target. And with that, I've used up most of my time and would like to acknowledge, as this was a large collaborative effort, all the different groups that contributed to this, especially our last publication, and of course NIH as the major funding source. Thank you very much, and I hope you have questions for me to answer.
Yes, this is Li Fan Lu. Hi, I have a question regarding to the memory that you talk about in terms of graft rejection. So when we talk about memory in the sense of infection, is usually when the antigen, the pathogen already clear.
Yes.
But in terms of graft rejection, the graft is still there. So,
correct.
So what is the purpose to have this innate memory if the antigen is always there? So why, when you disrupt the memory formation, then you can see the prolonged graft tolerance? Because, I mean, to me, that's not really memory in that sense, right? Because unlike the pilot study, when you actually immunize the splenocytes and then come back with a graft, but when you do the actual graft rejection study, the graft is always there. So can you comment on that?
Yes, I mean, this is, you're absolutely right. This is the situation we deal with in transplantation, and is very different from an infection or a situation where antigen will go away, right? So I think this is still important, memory or not. You're right. I mean, it's probably impossible to study memory in that setting, but you do have continued innate cell activation. So it is not, and this goes back to my initial statement, it is not that, so if only danger and microbial contamination would initiate the allo response, because that provides the second signal, and it's not, and the antigen of course comes from the graft, then you would think that that danger will go away at some point, right? But it's not. We know we get late rejections, we get chronic rejection. So we think that there is chronic activation of the innate immune system, be it memory or not, that leads to persistent generation of effector T cells. And in other work, we have shown that this is mostly happening in the graft at this point. Once you have effector T cells, they make continued interactions in the graft with these mono DCs that infiltrate, perpetuating the response in the graft locally. I don't think we're going to be able to show that there is local memory monocyte formation in that setting.
Yeah, I was wondering whether you were able to. I don't know, because clearly you show an impact, right? So clearly when you disrupt this pathway, the graft can be tolerated for longer time. So
yes.
so certainly they're doing something, but I don't know in terms of calling the memory. I mean, in terms of, in your first study, when you do the immunization first, that because they survive longer, so that's why the name.
Yes, no, I agree with you. It is certainly difficult to say that there is, we don't know. We will not be able to test if there is memory formation in the actual transplant setting over time, especially since I'm telling you that it is fairly short-lived.
Yeah.
But I think there is no doubt that there is such a thing as monocyte memory, and it is specifically allogeneic non-self.
Mm-hmm.
Of course, the bigger picture question is, why do we have this in the first place, right?
Yeah.
I know as a transplant immunologist, that immunologists always make the point that transplantation is not something that naturally happens. So in terms of what is the evolutionary pressure for this system to develop, that's, I think, a very interesting question. And I think Fatty Lack is going to investigate that further.
Okay, thank you.
Transplantation does happen, right? I mean, it's having a baby, placentation.
Yes, exactly.
So I think maybe
Yeah.
maybe looking at a placental model as a way to look at sort of tissue.
Yes, this is
memory.
exactly what Fatty's plans or interests are. Let's put it this way. Yeah.
I think I saw Elena had a hand up.
To be honest, I'm going to ask the questions about what would be the evolutionary pressure, not only for the memory monocytes, but for the polymorphisms of the SIRP alpha, and also what's the beyond the allograft rejection, right? What are the roles of what would be the evolutionary pressure of having such much polymorphisms in the SIRP alpha, and also the peer recognition of non-self MHC. What do you think are the roles beyond the allo rejection?
Beyond transplantation and placentation? I don't know. I think placentation could be a big evolutionary driver, but we don't really know. I mean, with respect to the peers, I know that SIRP alpha CD47 has a big interest in the cancer field. Although, of course, you can't make the point that it's a polymorphic SIRP alpha. It's more the presence or lack of CD47 or blocking that interaction that that has a more classical macrophage phagocytosis function. In that case, we have been thinking about viral infections, actually, not in terms of the SIRP alpha, but there are some indicators that the peer receptors could serve as entry molecules and could have developed as a response in terms of to immune evasion of virus and viral infections. So that's something that that I am trying to investigate further.
Thank you.
Do you guys have any idea where the pure molecules bind the MHC?
No, we don't. And I have to say, I think we were very lucky that we got the tetramer system to work. It's been really difficult because even the gene locus is so close together that we were only able to make a knockout for all 5 PRA genes. And there are no antibody reagents whatsoever, except for that one antibody that recognizes both PRA and PRB. So there are a lot of unknowns.
Right.
Can I ask another question? Yeah. So
of course.
So most of the data you show is the number of monocytes, right? So and early on you did have the data showing the IL-12 production. So
Yes.
My question is like with the SIRP alpha study. So in the PRA study, did they? So lack of this interaction, did they become more tolerogenic? So in terms of not just the number, but also whether they, for example, turn to promote regulatory T cell induction. Have you guys looked at that?
No, we haven't looked at that. The only thing we did look at was to make sure that the PRA knockout monocytes function normally. So we looked at cytokine production, TNF, IL-6, and so on. And we're able to show that during the primary response, they behave similar to wild-type monocytes. That was very important for obvious reasons to make sure that there is not that there are not additional functional impairments that have nothing to do with memory. But we haven't looked at what specifically happens to the memory response in terms of function when you block PRA.
Okay, thank you.
So thinking about the data that you showed with the differential expression of the peer A, B subtypes, it kind of reminded me of NK cell licensing. So is there a difference in strains as to which subtypes of peers are used or more common?
That's a good question. We are in the process of repeating our single cell analysis. And of course, we also want to look at epigenetic changes. And with that, we're hoping to expand it to more strains so that hopefully we can get a response to that. I mean, yeah, you will probably know that the analysis is not always straightforward in terms of, you know, which transcripts do you capture and which do you not capture. So with the 2 strains basically that we had, that we sequenced, it was really difficult to make any claims of that sort.
I think we're at the end of our hour, although I know you've been gracious and agreed to meet with a couple of the faculty members throughout the day. So I have a whole bunch more questions to harass you with later, but with that, I think we'll let you go and take a break here and then sort of meet with people individually. But thanks again. That was a fantastic talk and about a topic that I think a lot of people don't think about, which is innate immunity in transplantation. So thanks
thank
again.
you very much for listening.
Talk to you shortly.
Okay.
Thank you, Martin. Great
Thank
talk.
you.
Thank you